Hello, everyone. I'm Fan Zhu from the Bioinformatics Department in University of Michigan. We are the ring group for the Dream RE Challenge. Today, I'm going to show you our method using Gaussian process regression. So we started to participate in Dream challenges since last year, and we have won 4 sub-challenges so far. For the Dream RE challenges, we are the winner of the first sub-challenge, and we are the co-winner of the second sub-challenge. Our team, called Guan Lab, is formed of Yuanfang Guan and me. The rheumatoid arthritis primarily affects joints. It can result in painful and deformed joint, which can lead to loss of functioning. About 0.6% of the US adult population have RA. Women have 2 to 3 times risk as men. Anti-tumor necrosis factor or anti-TNF treatment is one of the most common treatments for RA patients. However, about 20% to 30% of the study population have failed to respond to the anti-TNF treatment. Here is a list of the 5 anti-TNF treatment we used in the RE challenge. I'll show you our performance in the leaderboard first. For the first sub-challenge, which is to predict the changes in the DAS28 score after the treatment, we are able to achieve a correlation of 0.54 with the target, which is highest across all the teams. For the second sub-challenge, which is to predict whether the patient is a non-responder, we are able to achieve an area under the ROC curve of 0.73 and area under the precision recall curve of 0.48, which is also the highest across all teams. For the final test set, in the first sub-challenge, we are able to achieve a correlation of 0.393. And for the second sub-challenge, we are able to achieve an area under ROC curve of 0.615 and area under the precision recall curve of 0.51. The method we used in this challenge is based on Gaussian process regression. And we have used 3 types of features. The clinical feature, the SNP features we extracted from the literature, and the SNP features we extracted from the training data. In the clinical feature, we found that the drug type is the strongest predictor. Then is the baseline DAS28 score before the treatment. We also found that the use of age, gender, and whether the patient has other co-therapies could further improve our performance. But that is not as strong predictors as the drug type and the baseline DAS. We didn't use the genotype batch or cohort information in our method because the final test set is based on a completely different one. We did a brief literature mining to generate a list of SNPs that is reported to relate either the risk of having the RA or the response of anti-TNF treatment. For each SNP in the list, we first evaluate using cross-validation in the training data, and we further evaluate using the leaderboard. So we only keep the SNPs that works in both cross-validation and the leaderboard. For the genetic feature we extracted from the training data, so we performed the cross-validation. We performed the correlation test, and there are lots of SNPs have a correlation larger than 0.2 to the target. But keep in mind that we have only 2,000 samples, but there are more than 2.2 million of SNPs. So we should expect that most of this correlation is coming from a rounding effect. So we end up selecting 10 to 15 SNPs from the literature, and only 5 SNPs from the training data. Our SNP set, which is proved to be successful in the final test set as well, can be considered as a robust one because it works in at least 3 independent data sets: the training data set, the leaderboard data set, and the final corona test data set. But the SNPs we selected either from the literature or the training data are much weaker predictors than the genetic features. So the method we used is called Gaussian process regression. It is not a simple linear or power or polynomial model. It can be considered as a much more complex version of the weighted mean method. It calculates using a covariance function to calculate the similarities between training samples and the target samples, and convert the covariance into weight. The additional benefit of Gaussian process regressions are it is robust to missing features, which is commonly observed in the clinical data. And it can also handle noisy data very well. And for example, the DAS28 score is a subjective score, so which is expected to be very noisy. So the noise tolerance of Gaussian process regression is exactly what we want. As the drug are the most important predictors, we first separate all the samples into drug-specific groups and build different models for different drug groups. In our final covariance function, the baseline DAS28 score contribute 40%. The age, gender, and whether this patient has other co-therapy together contribute 20%. And whether all the SNPs combined contribute the rest 40%. So in our model, 60% is coming from the clinical feature and 40% is coming from the genetic feature. We used exactly the same method for subchallenge 1 and subchallenge 2. And for the subchallenge 1, we used the changes in DAS28 score, which is a continuous value as the target. For the subchallenge 2, we used whether the patient is a non-responder, which is a binary value as the target value. With the help from Abhishek Pratap from Sage Bio Network, we are able to create the step-by-step provenance indicating how did we generate our final predictions from the source data, including the code and the intermediate files. And this provenance is now available on the Synapse website. Conclusion. So we built a Gaussian process regression-based model to predict the rheumatoid arthritis treatment response. And based on the result of DREAM challenge, I'd like to say that the Gaussian process regression is the best method to predict the rheumatoid arthritis treatment response. Here are some other thoughts for you to think about. So clinical feature is always the strongest predictors, while the genetic features are usually much weaker ones. So we need to keep this in mind when we adjust the weight between the genetic features and the clinical features. And Gaussian process regression seems to be a good one handling the genetic, weak features such as the SNP features from genetic. Okay, that's all. Thank you.